This lab is on mitosis and meiosis. And the cell cycle is when one cell becomes two, and there's 3 steps to this process. The first step is interphase, and in interphase all of the organelles inside of the cells are essentially doubling up. So like the ribosomes and the lysosomes and all those other somes are dividing. The second step is mitosis, or in the case of the egg and sperm, it goes through a slightly different process, and it's called meiosis. All the other cells in your body outside the egg and sperm are going through mitosis, and both of these processes involve only the nucleus of the cell. And the last step is cytokinesis. Cyto meaning cell, and kinesis implying some kind of movement, so it's the cell actually splitting, and some people consider that to be mitosis and cytokinesis to be the same process, and some people consider them to be different processes. So just to remind you, there's 4 or 5, depending on who's teaching it. There's 4 or 5 different steps to mitosis, and it's prophase, and then some books have prometaphase, and then there's metaphase and anaphase and telophase. Right, and so all you're doing in this lab is that you're looking at different microscope slides. I think the first lab involves the onion root tip, and so what you're going to see are a bunch of squares. It looks like a bunch of bricks next to each other, and then purple circles inside that. The circles inside are the nuclei, and so some of those nuclei are going to be regular and round, and some of them, and that's just a cell that has not begun to divide. Some of them are going to look kind of odd, like squiggly lines inside. Those are going through mitosis, right? And then the square part along the periphery, that's the cell wall. And when you look at the second part, which involves fish cells, which are animal cells, those are not going to have cell walls, so they're going to be shaped a lot different. They're not going to be packed in together. But nevertheless, you're going to see the nuclei going through different changes, at least those that are dividing. And so all this lab really wants you to do is look at these slides and try to determine, what is, what stage these cells are in. Most of them are going to be in prophase. That's probably the first thing you should keep in mind. And then the second thing you should keep in mind is there's no strict delineation between the phases, at least in my opinion. Where anaphase ends and telophase begins, that's all up for debate. So there's no definite black and white line, right? But you will know, you should at least be able to recognize the difference between prophase and like metaphase, for example, or prophase and anaphase. I mean, they definitely look different. And the introduction should show you some good pictures, or you can use the corresponding chapter in your book. So meiosis runs through the same process, but it does it twice because there's going to be some genetic recombination. You're going to have 23 chromosomes from the father and 23 chromosomes from the mother, and they're mixing together. And so the result of that is going to look different than both parents. Whereas in mitosis, the daughter cells look exactly like the original cells. So good luck with the lab.